When a tree fails and causes damage, the first question is never about the chainsaw cut. It is about foreseeability. Could the failure have been predicted? Could it have been prevented? That question is answered, more often than not, by how well the assessor understood tree biology and wood structure. Tree risk assessment is not defect hunting, it is biological interpretation. If you do not understand how wood is formed, how it functions, and how it loses strength, you cannot accurately evaluate likelihood of failure. And if you cannot defensibly evaluate likelihood of failure, you are not practicing within the intent of ANSI A300 Part 9. ANSI A300 Part 9 defines tree risk as the combination of the likelihood of an adverse event and the severity of the associated consequences. The adverse event in our context is failure of a tree or tree part impacting a target. Likelihood of failure is the probability of a tree or tree part failing within a specified time frame. That probability is governed by the interaction between load and resistance. Load is applied force: wind, gravity, snow, ice. Resistance is the tree's structural capacity to withstand that load without failure. Resistance is determined by wood anatomy, material properties, defect presence, decay progression, adaptive growth, and the tree's overall health and vigor. To understand resistance, we must understand what wood actually is. Wood is secondary xylem produced by the vascular cambium. At the microscopic level, wood is a composite material primarily composed of cellulose, hemicellulose, and lignin. These components form a biologically engineered structure optimized for vertical load support. Cellulose forms long, linear microfibrils aligned predominantly along the longitudinal axis of the stem. This alignment gives wood its tremendous strength parallel to the grain. Cellulose provides tensile capacity. It resists pulling forces and bending stresses. Lignin is the rigid polymer that surrounds and binds those cellulose fibers. Lignin provides compressive strength and stiffness. It resists crushing and buckling. If cellulose acts as reinforcing fibers, lignin acts as the rigid matrix that distributes compressive stress. When decay organisms colonize wood, they degrade these structural polymers. Brown rot fungi preferentially consume cellulose and hemicellulose. As cellulose is lost, tensile strength and bending strength decline rapidly, even when the wood may still appear intact. White rot fungi degrade lignin as well as cellulose, progressively altering compressive and tensile properties and often leaving a fibrous texture. Strength loss begins before visible cavities form. That is why biological understanding is essential when estimating likelihood of failure. Wood is anisotropic. Its strength properties differ depending on direction. Longitudinal strength, parallel to cellulose fiber orientation, is dramatically greater than radial or tangential strength. Trees are engineered to support vertical loads efficiently, not to resist forces equally in all directions. When longitudinal fibers are disrupted by cracks, seams, or decay columns, tensile resistance declines significantly. When cross-grain shear develops, structural integrity can be compromised quickly. Within each growth ring, Earlywood and Latewood differ in density and strength contribution. Earlywood, formed during rapid spring growth, has larger lumens and thinner cell walls. Latewood, formed later in the season, has thicker walls and greater lignin concentration. Latewood contributes disproportionately to structural strength. Growth rate alone does not determine structural quality. Material properties are influenced by environment, genetics, and resource availability. Trees respond to mechanical stress through reaction wood. Reaction wood is adaptive growth formed in response to persistent mechanical loading or displacement. In hardwoods, reaction wood is called tension wood. It forms on the upper side of leaning stems or branches and is rich in cellulose. Tension wood generates tensile forces as it matures, pulling the stem toward vertical alignment. In conifers, reaction wood is called compression wood. It forms on the lower side of leaning stems and contains elevated lignin concentrations. Compression wood generates compressive forces that push the stem upright. Reaction wood reflects prior load imbalance. It indicates the tree has experienced asymmetrical stress. However, reaction wood has altered mechanical properties and may influence future failure patterns due to non-uniform stiffness and internal stress distribution. Flexure wood develops in response to repeated bending stresses such as wind sway. Trees exposed to dynamic loading conditions develop wood with altered microfibril orientation and structural properties that enhance flexibility and energy dissipation. This adaptive response improves resilience over time. Now consider wounds. When a tree is injured, it produces woundwood. Woundwood is callus tissue formed around the margins of an injury. Over time, this tissue may roll inward and occlude the wound. Woundwood represents structural reinforcement. It is the tree's attempt to restore mechanical continuity. Pronounced woundwood ridges can indicate adaptive strengthening. However, bulging may also indicate internal decay expansion. Interpreting Woundwood requires biological context. The compartmentalization of decay in trees model explains how trees resist the spread of decay. Wall 1 resists vertical spread. Wall 2 resists inward spread through growth ring boundaries. Wall 3 resists lateral spread through ray cells. Wall 4 forms new wood that separates infected tissue from future growth. Compartmentalization is an active biological defense system. Its effectiveness varies by species and is influenced by tree health and vigor. This brings us to a critical but often overlooked factor in tree risk assessment: health and vigor. Health refers to the presence or absence of disease and dysfunction. Vigor refers to the tree's capacity for growth, resource acquisition, and physiological activity. A tree can be alive, yet have low vigor. It can be free of obvious pathogens, yet physiologically depleted. The ability of a tree to resist the spread of decay depends heavily on its health and vigor. Compartmentalization requires energy. Formation of chemical barriers requires resources. Production of woundwood requires active cambial growth. A vigorous tree with adequate carbohydrate reserves can form strong wall 4 barriers, produce substantial woundwood, and maintain radial growth that preserves residual wall thickness around defects. A stressed tree with depleted reserves may compartmentalize poorly. Decay columns may expand more rapidly. Radial growth may decline, reducing the addition of new structural wood. Over time, residual wall thickness may decrease relative to overall stem diameter. In practical terms, two trees with identical cavities may have different likelihood of failure ratings because one is vigorous and actively reinforcing the defect, while the other is declining and unable to maintain structural integrity. Crown density, shoot extension, leaf size, dieback, and annual growth increments provide insight into vigor. Soil conditions, root disturbance, drought stress, and site changes influence physiological capacity. When evaluating likelihood of failure within a specified time frame, we must consider not only the current structural condition, but also the trajectory. Is the tree maintaining itself? Is it producing sufficient new wood to offset decay progression, or is structural capacity declining year over year? Residual wall thickness is not static. It reflects the dynamic balance between decay expansion and radial growth. Health and vigor determine which side of that equation dominates. Load remains constant or may increase with crown expansion. Resistance depends on material properties, defect extent, adaptive growth, and physiological capacity. During a level 2 assessment, we observe defects, we identify targets, we estimate likelihood of failure, but our estimate is strengthened when we integrate structural biology with physiological condition. A cavity in a vigorous tree with strong compartmentalization and robust annual growth may present a lower likelihood of failure within the assessment time frame than a similar cavity in a stressed, declining tree with minimal radial growth. Advanced assessment may be justified when uncertainty about internal condition or decay progression materially affects management decisions. However, even advanced diagnostic tools do not measure vigor directly. That evaluation requires professional observation and biological understanding. Ultimately, tree risk assessment is the disciplined evaluation of whether structural resistance is likely to be exceeded by applied load, resulting in failure that impacts a target. Biology determines resistance. Cellulose provides tensile strength. Lignin provides compressive rigidity. Reaction wood redistributes forces. Woundwood reinforces damage. Decay degrades structural polymers. Health and vigor determine the tree's ability to resist decay spread and maintain structural capacity over time. Two trees with similar visible defects may have different risk ratings because their species characteristics, load exposure, decay type, adaptive growth responses, and physiological vigor differ. When you understand wood structure and tree physiology together, you move beyond static inspection. You begin assessing a living structural system in motion. Tree biology is not a peripheral concept in risk assessment. It is the foundation. Without understanding health, vigor, and structural material science, likelihood of failure becomes assumption. With that understanding, likelihood of failure becomes informed, defensible professional judgment grounded in the biology of living wood. Thanks for listening to the Arborist Podcast. Let's keep building your arborist knowledge one domain at a time. If you found this useful, please like and subscribe.